Hello guys. So for your CSIR net exam today we will revise apoptosis. This is in unit 4. Okay, this comes under unit 4 and let's begin the quick revision of the topic apoptosis. So apoptosis is a kind of programmed cell death, and necrosis is what? Necrosis is a death of living tissue. So non-apoptotic death is necrosis, apoptotic death is kind of both are programmed cell death. Necrosis is kind of not programmed. If it is programmed necrosis, then it is necroptosis. So when does cell die? So when there is no control over the cell division, then what happens? Cell keep on developing, keep on dividing. So controlled cell death is needed for normal development. Okay, for good health, for normal development, controlled cell death is required. And this cell death may occurs in many ways, maybe infection, poisoning, lack of oxygen. And this uncontrolled cell death, it is messy. Okay, so the cell swells up and the contents leak away. In that case, what happens? It is very damaging for the cells, so there must be a cleaner way for the cell death. Okay, this is the programmed cell death. So when does apoptosis occurs? It occurs during viral infection, during DNA damage to make new cells. In the womb, our fingers and toes, they are connected to one another by a sort of webbing when we were born, but when we were like developing in the fetus. But apoptosis occurs between the fingers, so that the gap like forms. Okay, so that a digit separates. So we are just revising, so I'm going a little quicker. Also, programmed cell death is needed during menstruation, and this is very important for uh, the cell turnover and proper development and functioning of immune system as well, embryonic development as well. So this programmed cell death also occurs in nematodes, C. elegans as well, and around 1090 somatic cells are generated in the formation of adult worm, of which 131 undergoes apoptosis. Okay, so many undergoes apoptosis in case of C. elegans, and these cells die at a particular point during development. This is necessary for accuracy and control in its system. So this apoptosis is very necessary in both plant and animal tissue development. And what if apoptosis stops? In case when anti-cancer drugs or radiation, what they do, they trigger apoptosis in disease cells. So if there is apoptosis which is stopped, and there will be uncontrolled cell division which will lead to cancer. Okay, so it is a characteristic of AIDS, Alzheimer disease, Parkinson disease. So these disease would be seen when apoptosis would be stopped. Autoimmune disease will occur, improper cell cycle. So how apoptotic bodies are removed by macrophages? How are they removed by macrophages? So the removal of dead cells, they can be done via phagocytosis. In living cells, the phosphatidylserine, which is found in the inner side of the plasma membrane, the protein named flippase, it maintains the phosphatidylserine in the cytosolic phase. But in dead cells, this is flip flops to the outer side, and this marks the cell for phagocytosis. When the phosphatidylserine goes towards the outer side, it marks the cell for phagocytosis. The macrophages bind to this phosphatidylserine, which is exposed in apoptotic bodies. Okay, and there are some phosphatidylserine receptors on MHCs. So first of all, there is a recruitment to the find me signals. Okay, so apoptotic cell have some find me signals which recruited macrophages, then recognition of newly exposed eat me signals and engulfment of the corpse. So there are some eat me signals which is PS. This will help the macrophages to bind. Then there is processing and degradation of the corpse. So post engulfment consequences, it releases some kinds of anti-inflammatory cytokines. So there are three pathways of apoptosis, three mechanisms: extrinsic, intrinsic, and perforin. So there are proteins named caspases, which are endoproteases. They have active cysteine sites which cleaves C-terminal side of the aspartate residue. Okay, caspases is C-terminal aspartate cleaving proteases which cleaves the C-terminal site of aspartate residues, and their catalytic activity depends on some cysteine residue enzymes. Okay, their catalytic activity have pentapeptide. Okay, on the cysteine residue with highly conserved active site consists of pentapeptide, which is QACRG, glutamine, alanine, cysteine, arginine, and glycine. So 7 different caspases have been identified in Drosophila and 14 has been found in mammals. 7 in Drosophila, 14 in mammals, where caspase 11 and 12 are the ones only identified in mouse. So these caspases, they form a cascade of proteases which are activated in this process. Caspases are formed as procaspase, which later on becomes activated into caspases by some apoptotic inducers. So these caspases, they are family of protease enzyme. So more than 12 caspases are being identified and they contain cysteine residue in their catalytic side having pentapeptide, which cleaves C-terminal of the protease. So different types of caspases are there: initiator caspase and effector caspase, executioner caspase. So what are initiator caspases? Initiator caspases are 2, 8, 9, 10, which are self-activating, and effector caspases are like activated by initiator caspase. So these are we discussed. So procaspase, they split into small and large subunits. Okay, small and large subunit, you can see in this picture procaspase split into small and large subunit. The small subunit and large subunit form heterodimer. Okay, this heterodimer uh, will form two such dimers and it will form active caspase. So this is active caspase. So these caspase, they are different phases of apoptosis. One is initiation phase, another is signal transduction phase, and the third is execution or death phase. Three phases are there: initiation phase, signal transduction phase, execution or death phase. So let's discuss about the three pathways one by one. So in the initiation phase, what happens? There is the intrinsic or extrinsic pathways activated by a stimulus. In case of signal transduction pathways, there is the activation of caspase and certain kinase phosphatases are involved. Initiation phase, where intrinsic or extrinsic pathways are activated. Signal transduction phases, where there is some caspase or kinase activation is involved. Execution is effector caspases are involved. So initiation, transduction and activation is there. So first of all, there is a healthy cell. There is some error which results from injury. So cell tries to repair itself. So if there is any additional injury to the cell, and there is the cell is unable to repair and cell enters apoptosis. So coming to the intrinsic pathway of apoptosis, if there is severe DNA damage, in that case, there are some pro-apoptotic factors which will be formed, like for example, back, okay. Now this backs will create pores on the mitochondrial membrane. This backs, pro-apoptotic, it will create pores on the mitochondrial membrane, as a result of which cytochrome C will be released in the cytoplasm, after which apoptosome will be formed. Okay, this cytochrome C will interact with APAF1. Cytochrome C will interact with APAF1 and form apoptosome. Apoptosome will, what it will do, it will react with procaspase 9 and form caspase 9. Caspase 9 will activate procaspase 3 and form caspase 3. So 9 is initiator and kind of 3 is your executioner. So it is non-receptor mediated signaling in intrinsic pathway where mitochondria is involved. So there is some stimulus which is required, say for example viral infection, damage of DNA by toxin, free radicals or radiation. There is damage to the DNA which induce apoptosis, and pro-apoptotic proteins, they activate caspases that mediate cell destruction in many ways. Okay, and these pro-apoptotic proteins, they translocate to nucleus including DNA fragmentation. So these pro-apoptotic events, they occur through the member of BCL2 family member proteins and tumor suppressor protein P50. So this pro-apoptotic event, it is regulated by two factors. One is BCL2 family and another is P53. So what do? This members of BCL2 family, they may be pro-apoptotic or anti-apoptotic. Okay, so there are some anti-apoptotic proteins like BCL2X, XL, BCL, BCLW bag. So mainly all BCL containing ones, two which are anti-apoptotic, and BCL10 is pro-apoptotic like BAX, BAC, BID, BAD, BIM, BIC. So all these are pro-apoptotic. So if there is DNA damage, there will be some abnormalities, say for example hypoxia. In that case, P53 is released. This P53 will either show apoptosis or it will show cell cycle arrest. In cell cycle arrest, there will be repair, and in apoptosis, there will be death. So intrinsic pathway, it is triggered by some stimuli, and here mitochondria plays important role. Cytochrome C binds APAF1 forming apoptosome, and this apoptosome activates procaspase 9 to caspase 9, which will activate procaspase 3 to form caspase 3. It requires DATP. So these mitochondrial proteins, they are known as SMACs, small mitochondrial derived activator of caspases. They are released into the cytosol following increase in permeability. So one is cytochrome C which comes out, and another is small mitochondrial derived activator caspase. They also come out from the mitochondria. These are the caspase activator. So this permeability transition pore, they induces what? Translocation and multimerization of pro-apoptotic protein bags. So this permeability transition pore, the pores which are formed, what it will do, it will help in translocation and multimerization of some pro-apoptotic proteins, so that uh, there will be leakage in cytochrome C and other proteins which give signal to apoptosis. So this spore helps in translocation of pro-apoptotic proteins so that cytochrome C can leak out. And this SMAC, small mitochondrial activating caspases, this is a mitochondrial protein, and this inhibits the it inhibits the inhibitor of apoptosis protein, which is IAP, inhibitor of apoptosis protein. So SMAC and below, what they are doing, they are inhibiting the inhibitors of apoptosis. What is inhibitor doing? Inhibitor is blocking procaspase 9 and 3. Another one is HTA2 and OMI. They also inhibit IAP. So IAP is inhibited by SMAC, Diablo, HTA2, OMI. All are inhibiting IAP. So there are several elements of ROS which is also included, such as the formation of highly toxic nitrogen reactive species. So caspase 3, once it is formed, it induces endonuclease such as caspase induced DNAs. Okay, caspase 3, it will induce endonuclease such as caspase induced DNAs. Caspase 3 will induce some endonuclease. So what happens? First of all, there will be DNA damage, free radical ROS, calcium ions, they will be released, which causes the dimerization or multimerization of BACs. BACs dimerization formed pore in mitochondria. It is forming pore in mitochondria. So first Bax comes into role. Bax, what it does, that forms spore. Once spore is formed, okay, uh, so once spore is formed, membrane potential is disturbed. Membrane potential is disturbed. So when membrane potential is disturbed, cytochrome C is released. Cytochrome C plus FF1 plus DATP, they all form apoptosome and they combine with procaspase 9. Okay, uh, so procaspase 9 along with cytochrome C and apaf1 is known as apoptosome. They activate procaspase 9 and form caspase 9, and caspase 9 convert procaspase 3 into caspase 3, which is the executioner caspase. And there are some inhibitors which block the inhibitor of apoptosis, which is IAP. So this was about intrinsic pathway. So if we discuss the steps again, first of all, Bax polymerization, internal signal is there. There is anti-apoptotic protein should be inhibited. Apoptotic protein multimerization will be there. Oligomerization or dimerization or multimerization will be there. Back formed. Back what it will do? It will form pore. Cytochrome C, APAF1, procaspase 9 will combine to form apoptosome. Caspase 9 will be formed, caspase 3 will be formed. Caspase 3 is the executioner caspase. So pro-apoptosis protein are Bax, BAD, BID, etc., Puma, Noxa, Trail 1, Trail 2. Anti-apoptotic are BCL2, XLW, MCL1, A1, BCLB. So all BCL which contains, here BCL2 will be there, pro-apoptotic. Here BCL10 will be there. Did I tell right? Let me check. BCL2 is anti-apoptotic. BCL10 is pro-apoptotic and BCL2 is anti-apoptotic. Okay. So ultimately what we see in intrinsic pathway, there is caspase 9 and then 3 and execution. In extrinsic pathway, there is a death ligand and death receptor involved. Adapters are formed which are linking to another proteases. There is a disc formation and caspase 8 activation occurs. In granzyme pathway, perforin and granzyme B is involved. Caspase 10 activation. So here it is 8. Here it is 9. So easy. Here it is 10 and all are activating caspase 3. Now coming to execution path, extrinsic pathway. In extrinsic pathway, there is a ligand which is attached to the apoptotic cell, and there is a receptor which is attached to, say for example, macrophages. Okay, so there is a disc formation, that there is a receptor which attaches itself to the which attaches itself to the domain. Some domains are there in the receptor, and these domains are uh, these domains are binding itself to procaspase 8. Procaspase 8 will form caspase 8. So what are the kind of ligand here? Ligand is TNF receptor family, FAST, DR5, DR4, TNF, and this is like GSK3. So what happens here? Say for example, in killer lymphocyte, there is extrinsic pathway. Okay, extrinsic pathway where ligand FAS cell binds to the receptor. Receptor have some domain known as death domain, FADD. If this death domain recruits itself on it some caspases. like procaspase, which gets activated into caspase 8. So there is some extracellular signal, that is ligand, which ligand and receptor binds with each other. There is a domain recruitment. There is a domain recruitment, death domain recruitment. Then procaspase 8 activation into caspase 8. There is a disc formation. Disc formation means caspase 8 when it is uh, activated and still like kind of they all are bonded to each other. Okay, then activation of executioner caspase, which is caspase 3, then apoptosis. So the ligands already discussed, it can be TNF, fast ligand, cytokine. Receptors can be cytokine receptor, TNF receptor, fast receptor. Okay, so instead of FADD, it can be TADD domain as well. Okay, both are same. So when it is TNF alpha, it is TADD domain. So extrinsic pathway is what? Extrinsic pathway here is that the signal is outside the cell, and there is a binding of the ligand, and the death receptors belong to the tumor necrosis receptor superfamily TNFR. And one typical example is fast receptor, another is TNF receptor. So these receptors, they have cysteine rich extracellular domain and a death domain which is 80 amino acid long region. So this death domain, they transfers the signal from the cell surface to the intracellular signaling pathway. Okay, the most studied receptor are fast, trail R1, trail R2. Ligand is fast L, which is homotrimeric in nature. This ligand is homotrimeric in nature, and it is transmembrane protein carried out in killer lymphocyte. So when this receptor binds to the ligand, there is a adapter protein called FADD, which is fast associated protein with death domain. So this protein have a death domain which is fast associated. This is an adapter protein which becomes which is associated with fast receptor. Okay, this death domain, which is FADD, it induces signaling complex formation known as disc. So there is a downstream signaling cascade which is activated. It will start and induce make caspase 3 active, and before caspase 3 activation, caspase 8 is activated, and the death receptor induced apoptosis is inhibited by a protein named as C-flip that binds to FADD and caspase 8. So there is a protein. So C-flip or FADD and caspase 8 which inhibits the uh, death receptor. So you can see here that it is this is homotrimeric. Here it is not shown properly. The ligand is homotrimeric, and there is an adapter protein which is FADD. It has death domain. Death domain, when combines with procaspase 8 and forms caspase 8, it forms disc complex. Okay, and some executioner caspases will be active, which is 3, 6, 7. Okay, so this extrinsic pathway, they involves receptor mediated signaling. So if cell is no longer, so where will you see receptor mediated signaling, extrinsic or intrinsic? You will see receptor mediated signaling in case of extrinsic pathway. Okay, so if cell no longer needs or diseased no longer needed, or the cell is diseased, it induces apoptosis. There are some chemical messengers like fast, trail, TNF. They may be excreted by some neighboring cells if the cell is damaged or no longer needed. Okay, and when there is a change in intracellular domain of this kind of the inducer, kind of the proteins which gets attached to the FADD, a protein inside the cell called FADD also changes. So if some proteins which binds to adductor, if those proteins changes, then a protein inside a cell called FADD also changes. So FADD interacts with other proteins to start the process. Okay, so if there is a change in intracellular domain, if there is a change in some domain of FASR or trailer, in that case what will happen? In that case what will happen? So if there is any change in this domain, trail and this TNF, in that case this depth domain also changes. The domain in FADD also changes. So that FADD can interact with other proteins downstream to start the process. Okay, and so if two of these molecules encounter an activated FADD, what will happen when the FADD is activated? Then the parts of the proteins that keep them inactivated are cleaved or cut away. Okay, so if FADD is activated, uh, then what will happen? The part of these proteins that makes these kind of protein inactive, like for example, if procaspase, in procaspase some part is cleaved off to form caspase. So that part is cleaved off when they are going to bind with, when they bind with FADD, when they are binding with active FADD, their inactive part is cleaved off to form active protein. Okay, so these active ones, 8 or 10, once they are active, they disperse to the cytoplasm and trigger several changes throughout the cell. Okay, say for example, breaking down of the DNA. So another inactive molecules called BID. BID is what? Pro-apoptotic. It is transformed into T-bid when the activated caspase cleave off the part of the bid that keeps the molecule inactive. Okay, so when the caspase is active, it forms bid to T-bid. T-bid is kind of, you can say, it's the cleaved bid. Okay, cleaved bid is active one. And how this bid is cleaved? It's cleaved by activated caspase. So after bid is transformed into T-bid, or T-bid moves into the mitochondria and activates the molecule, kind of permeabilizing the mitochondria. Another one, backs back. So when backs and back are activated, it will create the pores. So what is the role of bid? Bid will activate some molecules in the mitochondria. What is the role of backs back? It will create pores in the mitochondria and induces some intrinsic apoptosis. So you can see here the difference between intrinsic and extrinsic apoptosis. And extrinsic is receptor mediated. Intrinsic is intracellular, where cytochrome C is released. So in here, extracellular caspase 8 is activated, and in intracellular caspase 9 is activated first. The third pathway is perforin granzyme pathway, where there is an interaction between cytotoxic T cells CD8 plus and infected cell. It is also a type of intrinsic pathway where cellular components are involved. There is no ligand receptor concept. So it is receptor mediated, independent of receptor mediated. So what happens here? That perforin is what? Perforin is a protein which forms pore. Okay, and granzyme is what? Granzyme is a family of related, structurally related serine proteases which are stored within cytotoxic granules. Okay, they are kind of serine proteases which are stored in the cytotoxic granules of cytotoxic lymphocytes. Okay, one is pore forming and one is serine protease. Okay, this perforin, what it will do, it will release granzyme B or granzyme A. Granzyme B will form will activate caspase 10. Granzyme A will activate. Granzyme A is caspase independent. It shows DNA cleavage. Okay, so perforin will form the pore. Granzyme, what it will do, it will activate the procaspase 8 into caspase 8, as a result of which there will be macrophages which will come and it will bind with this signal. So how apoptosis is regulated? So there are some inhibitors of apoptosis, which is IAP, inhibitor associated proteins, inhibitors of apoptosis. They are the regulators of apoptosis. Okay, and the mammalian family of BCL2, the mammalian family of BCL2 family of anti-apoptotic genes, they are the homologue of CED9. So CED9 is anti-apoptotic. CED9 is anti-apoptotic, which is found in C. elegans. Okay, and BCL2 proteins is what? This is homologue of CED9 and inhibits apoptosis, and backs back, which forms the pore, while BCLX inhibits it. BCLX is what? BCLX is anti-apoptotic or pro-apoptotic. BCL2 is anti-apoptotic. Okay, but BCLX is what? So BCLX is what? It is the anti-apoptotic one. BCL2 is what? BCL2 is pro-apoptotic one. So this apoptosis is also regulated by HSP, which is induced by stress. So HSP, what it does? It inhibits apoptosis. So what are the inhibitors of apoptosis? IAP, then BCL2, BCL2, BCLX, okay, then HSP. Then HSP90 also blocks the oligomerization of APAF1, inhibiting intrinsic pathway. 27 and 70, they block the signaling pathway of TNF alpha or FAST receptor. Okay, HSP90 blocks APAF1, HSP27 and 70 blocks TNF alpha or fast. That means HSP27 and 70, what it is doing, it is inhibiting the extrinsic pathway. HSP90 is inhibiting the intrinsic pathway. HSP6010 helps in activation of caspases. So some are inhibitors, some are activators. 6010 are activators. So there are several known inhibition of apoptosis proteins, which is uh, kind of inhibitors of apoptosis. What they are? BIR 1, 2, 3, 4, 5, 6, 7, 8. They are the inhibitors of apoptosis. So there is a cell death pathway in C. elegans as well. So programmed cell death is C. elegans is also observed by, also some homology of these sequences is observed. So around 2 dozens of the genes are involved, which helps in the cell suicide program, and it was found that these genes are conserved, and these genes, they play a very important role in apoptosis in nematodes like CD3, 4, 9 and EGL1. So human BCL2 was what? Human BCL2 was anti-apoptotic, and this is homologous with CED9 protein of C. elegans. But sequence wise, very little symmetry is there. So if BCL2 gene is like introduced into CED9 mutant form, apoptosis pathway will suppress. So this means that BCL2 and CED9 have similar role. So initiator caspase in case of mammals it was 9 and effector was 3 or 7 in case of intrinsic pathway. Okay, in case of C. elegans the effector caspase is caspase 3 or CD3. Okay, CD3 is the effector caspase, and the pro-apoptotic protein in case of C. elegans is EGL1, which is back. It is EGL1 in C. elegans, and pro-apoptotic protein CED4 is also found in C. elegans. While in Drosophila, you'll find Reaper, Hid, Grim are pro-apoptotic protein, and Diap1 is anti-apoptotic. Dronk is caspase 9 like. Okay, caspase 9 is what? Initiator caspase. Dryce is caspase 3 like. DCP1 is caspase 3 like. So when we try to understand about C. elegans, EGL1 is what? EGL1 is pro-apoptotic. Okay, CED9 is anti-apoptotic. Okay, CED4 is what? CED4 is, say for example, it is kind of similar to kind of working in intrinsic pathway. Okay, and CED3 is the executioner caspase or caspase 9 or caspase 3. In mammals it is caspase 9 or caspase 3, and in C. elegans it's CED3. So in C. elegans, CED4 and 3, 4 and 3 execute the events of cell death. CED4 and CED3, they execute the events of cell death. Okay, CED4 execute CED3, 4 and 3 executes the process. CED9 is the inhibitor. So they are inhibited by CED9. So CED9, they inhibits CED4 and inhibits CED3. And this inhibition is lifted by expression of EGL1. What is EGL1? EGL1 is pro-apoptotic. EGL1, it is inhibiting CED9. This is pro-apoptotic. Another one is TRA1 and CES1. They are the transcription factor that inhibit expression of EGL1. So EGL1 is again inhibited by TRA1 and CES1. Okay. So EGL1 inhibits CED9. CED9 inhibits CED4. CED4 and 3. CED4 are what? CED4 and 3, what they do? They helps in apoptosis. In mammals, what happened? There is bid backs, they are pro-apoptotic. Okay. SMAC is what? SMAC, IAP, they all are inhibitor of apoptosis. SMAC inhibits IAP. Sorry, I told wrong. SMAC is pro-apoptotic. SMAC is kind of inhibiting IAP. Cytochrome C, what it is doing? It is the intrinsic pathway. And extrinsic has been cut off mistakenly. So EGL1 is what? It is pro-apoptotic. BCL2 member. CED9 is anti-apoptotic. CED3 and 4, they are kind of, you can say, executioner caspase. So we already discussed that there are two forms of cell death. Necrosis is what? There is no programming required. If programming is there, is necrosis. It is necroptosis, where cells swell and there is some inflammatory substances also released. cell rupture occurs, there is no membrane blebbing, but all of these are seen in apoptosis where macrophages play an important role. Cell adhesion is lost, cell shrinks, there is membrane blebbing, fragmentation. So this was all about apoptosis, where you have understood about the three pathways of apoptosis, and you can see that here blabbing occurs. Blabbing is like kind of ridges formed in cell membrane, which forms which breaks the cell into distinct bodies. Okay, the organelles are still functional until these are broken down by macrophages. Okay, so the blebs fuse and become larger. No organelles are located in the blebs. Okay, in case of necrosis, what we see that small blebs, if it forms, the structure of nucleus changes. There is no fragmentation of the nuclear wall as such. So these blebs fuse and become larger, and no organelles are located in the bleb. But here organelles are located in the bleb. The cell membrane ruptures and releases the content, and the organelles are not functions. Here no content is released. So this is all about apoptosis. If you have any questions, you can ask in the comment box, and I hope this will be very helpful. And from here you can expect all the questions, and no questions will come outside of this topic. This is very helpful for your all competitive examinations. Thanks a lot for watching till the end. Thank you so much. Don't forget to subscribe the video. Like and subscribe motivates us a lot for making further videos. Thank you.